Welcome dear students. Today we will study about structure of a bacteriophage and that of HIV virus. Now why do we call it bacteriophage? It is called so because it is a bacteria eater virus, or it attacks on bacteria. Student, it is tadpole in shape. Simply it can be called as what? Phage virus. It consists of three parts: head, neck and tail. Student, head that consists of outer part which is called as what? Capsid, or also called as what? Coat of the head, and inner core that consists of single stranded DNA. Now a head that is icosahedral in shape, and icosahedral shape is a shape which consists of 20 triangles. How many triangles they are there? 20 triangles they are there in icosahedral shape. And student, neck that connects head with that of the tail, and tail that is a hollow tube, and it is externally surrounded by a contractile sheath which helps it to transfer a viral genome into that of the host cell or bacterial cell. This tail that ends up into a basal plate which consists of 6 tail fibers and number of tail pins. Both tail fibers and tail pins, they are used to attach the bacteriophage with that of the host cell. Student, at the end of the tube, an enzyme it is released, which is called as what? Lysozyme. And upon the contraction of this tail, and this lysozyme that dissolves bacterial cell wall, as a result it can transfer its DNA into that of the bacterial cell. Now next is HIV virus. HIV is spherical or rounded in shape. HIV virus is human immunodeficiency virus. It consists of outer envelope which consists of lipid bilayer along with some proteins which are glycoproteins. Student, these glycoproteins they make up spikes, and with the help of these spikes an HIV virus it can attach itself with that of the host cell. These glycoproteins, they are of two kinds. One is GP120, which is also called as a surface protein, while the other one is GP41, which is a transmembrane protein. Both the proteins they are used to attach and to fuse the HIV virus with that of the host cell. Next part is a matrix protein. Matrix protein is a part or is a layer which is present between the envelope and that of the capsid. Students, in HIV virus, capsid that is a bit conical in shape, and it consists of what? Proteins, and while its inner core that consists of two RNA molecules. Along with RNA molecules, it is having different enzymes which are reverse transcriptase, integrase and protease. Student, reverse transcriptase that is involved in reverse transcription means it converts these RNA molecules into DNA molecules. Process is called as what? Reverse transcription, because you know transcription is what? Making of RNA from DNA, but when DNA that is being made from RNA, then this process is called as what? Transcription, sorry, reverse transcription. Another enzyme is integrase. This enzyme that binds or attaches DNA of the HIV once it is formed into that of the host DNA. That's why it is called as what? Integrase, because it is involved in integration. Third enzyme is called as protease. Protease enzyme that breaks different proteins into smaller fragments, and these proteins of the viruses which are like GP proteins, matrix proteins and these capsid proteins, they are encoded by 3 genes of the HIV virus. Rest of the proteins, rest of the genes, they are involved in making some regulatory proteins which are different enzymes. So these 3 proteins, they are what? Structural proteins, and they are encoded only by 3 genes. So this is all about what? Structure of HIV virus. Thank you, students.